Hi, good afternoon and welcome. My name is Brian Roberts. I'm the Associate Scientific Director for Protein Therapeutics at AAPS Inc. in Providence, Rhode Island, and I oversee the immunogenicity screening services for generic and novel peptide drugs and their impurities. Welcome to my talk today entitled Addressing Immunogenicity of Impurities for Peptide-Accelerated New Drug Applications. And thank you for listening today and for the organizers for giving me the opportunity to speak. These are our session description and objectives, and my biography and contact information. Should you have any questions about this talk or our screening services in general, I'd be more than happy to talk to you about those. So as many of you are probably aware, back in 2017, the FDA released a draft guidance entitled ANDAs for certain highly purified synthetic peptide drugs that refer to listed drugs of rDNA origin. And in this case, as generic drugs become more widely available on the market, synthetic peptide manufacturing is being used to produce these drugs as opposed to the recombinant DNA technology. And with synthetic peptide manufacturing, there's always the risk that impurities could be present that result from failures in the manufacturing process, or also degradation impurities from storage and conditions. And both of these types of impurities can actually lead to new T-cell epitopes that might be present in the product, and some of these impurities that could raise the immunogenicity profile of the generic product over and above that of what was known for the reference product. And so at Epivax, we've been doing the screening for over 20 years, both for biologics and vaccines. And so we took peptide, and which these drugs are peptide drugs, and combined that with ANDA to come up with our Panda process, and that's why you see this little guy up here in the top corner of my slides. The Panda Approach, or Peptide Accelerated New Drug Application Approach, is a three-step method that we use to evaluate peptide drugs and their impurities that we can use to get a very full picture of the risk posed by the product and any impurities that might be in it. And it starts off with an in silico analysis of both the API sequence and any of the impurities that you've identified that are within or above the limits spelled out in the ANDA guideline. Any peptides that we predict to bind in silico are validated in our in vitro class 2 binding assays, and if we've confirmed binding from what was predicted in silico and we do see an increased risk of immunogenicity associated with that, then we move into these ex vivo T-cell assays where we look at the ability of those peptides and impurities to generate a T-cell response, which can ultimately lead to an anti-drug antibody response. And from that, we can look at the current literature that's out there about your product and the reference product, and we can combine all this data together and put in, compile a full immunogenicity opinion report that can be used to be submitted with your ANDA application. So as a part of the ANDA guidance, the FDA became interested in knowing how in silico tools and in vitro tools could be utilized to look at the risk of impurities in peptide drugs, and we applied for a contract to show them our tools, and which we were awarded. And during the course of that contract, we looked at two peptide drugs, salmon calcitonin and Teraparatide, and their impurities. And what we hope, based on that analysis, and what we hope to show you today, is that in silico analysis can tell you what to look for in an in vitro assay, and this is based on the nature of the API itself and the impurities as well. And really what we're looking for is, are those impurities in the API immunogenic, are they tolerogenic, or are they null? An immunogenic API, such as salmon calcitonin, is likely to have very immunogenic impurities, and the impurities are not likely to be more immunogenic than the API itself, especially at the concentration that they're usually present within the final drug product. So the API in this case will actually overshadow any immunogenicity that's found from the impurities. However, when you take a drug like Teraparatide, which has low immunogenicity and is actually thought to be tolerogenic, that drug itself, or API itself, will have low immunogenicity, which we can address and identify in silico. And that tolerogenic nature is due to the homology that peptide has with several cell proteins. And if you break that tolerance, however, any impurities that have a loss of tolerance or loss of homology are likely to be more immunogenic, and if present at high enough levels can actually impact the immunogenicity of the overall product. And so combining at least two of the three possible tests in silico, in vitro binding, and T-cell assays are very useful approaches for preparing your ANDA submission and evaluating your drug product. So we're going to look at, as I mentioned, two peptides, salmon calcitonin and Teraparatide. So today we're going to have a tale of two peptides. We have the best of times and the worst of times. So both of these drugs are used to treat osteoporosis. They do share some common features, but at the same could not be more different in terms of their immunogenicity. So Teraparatide is human-derived and is the N-terminal 34 amino acids from parathyroid hormone. It has a history of being non-immunogenic in clinical trials, so only about 3% of the patients developed anti-drug antibodies to teriparatide, whereas salmon calcitonin is very foreign, and as the name implies, it's derived from the salmon calcitonin product peptide, and this peptide only shares about 50% sequence homology with human calcitonin. 40 to 70% of the patients who have been treated with this drug developed anti-drug antibodies in clinical trials after a couple months of treatment. Both of these two peptides contain a feature that we identify in our in silico analysis called an epibar. An epibar is a 9-mer peptide frame that is predicted to bind 4 or more of the HLA supertype alleles, and we'll show you that in just a second. While they both have this epibar, we believe that the teriparatide epibar is actually promoting tolerance due to its cross-conservation with the human genome. And however, the salmon calcitonin one is in about the most foreign region of the peptide compared to the human calcitonin, and in this case we think it's driving immunogenicity. So here's salmon calcitonin we'll start with today, and here's our epimatrix cluster report. So this is our in silico analysis, and what you can see here is all of these shaded boxes are predicted binding. The darker the box, the more significant we think that binding interaction with these supertype alleles, which are shown up at the top, will be. And you can see with salmon calcitonin, most of the epitope content is located within the C terminus. And as we mentioned here, this is the epibar in frame 16. And this epibar, these 9 amino acids, 6 of the 9 amino acids differ from human calcitonin, so this is a very foreign peptide in a very foreign region, and because of that we believe that this is what is actually driving immunogenicity, and some early data on salmon calcitonin supports that, as well as the clinical observations of 70% immunogenicity in patients. We have what's called our Janus matrix human homology score, so anything below 3 is considered to not share homology with the human proteome, and as you can see here, this is a very low potential for tolerance. When we were provided with 4 impurities that were identified in generic salmon calcitonin, and when we look at these, you can see where the modifications leading to the impurities are. They're highlighted in red here, so it has to do with an acetylation, a change of amino acid, or insertions at different spots. What you can see is the overall number of epitopes or epimatrix hits remains the same between these impurities compared to the API. We see very little change in the actual epimatrix immunogenicity score, which is largely based on epitope content, and we see very little change to the human homology score. So if anything, some of these peptides become slightly less human-like. So as you can see that these, in an immunogenic and very foreign peptide, any modifications leading to these impurities are unlikely to significantly change the predicted immunogenicity of those impurities over that of the API itself. When we look at these peptides, then in vitro in our T-cell assays, we won't be showing you the HLA binding data today due to time, but we looked at this in 2 different ways. So we did a comparison of the individual peptides, so the salmon calcitonin peptide compared to the impurities. And what we see is when they're all compared at the same concentration, these peptides themselves, or the impurities themselves, are slightly more immunogenic than the actual the API sequence. So we do see a higher potential for these to be immunogenic. However, to test to see what impact they might have on the final drug product, we took myocalcin, which is the reference product for salmon calcitonin, and we actually spiked myocalcin with these impurities individually at their observed percentages as they were identified in the generic product. And what you see here is that there's very little impact on the immunogenicity compared to myocalcin. So while these impurities are all have the potential to be slightly more immunogenic when compared at the same concentration as the API, when they're there in a much lower concentration or the observed concentrations, you actually don't really see much of a difference. These responses were lower than expected to begin with, and this may have been due to one of the excipients within the formulation, and I provided a talk earlier last week that describes this in more detail, so I would encourage you to check that out if you're interested. So the results of the IVIP T-cell assays confirm that in silico results predicting that both the API and the impurities will be more immunogenic. Immunogenic peptides will produce immunogenic impurities, and in these assays, the immunogenicity of the API overshadows that of the impurities in the final drug product, likely due to their concentration within the final drug product, and ththese impurities don't make the RLD more immunogenic when present at their observed abundances. So the amount of impurity, in addition to its immunogenic nature, relative to the API, can determine their contribution to the overall immunogenicity of the final drug product. Now switching over to Teraparatide, which is a different story, we can see here that again, we have this epi bar within salmon calcitonin, or within Teraparatide. You'll see that we have an elevated epi matrix score compared to that of salmon calcitonin, and that's likely due to the fact that there's just more hits or predicted T cell epitope content than there was for salmon calcitonin. This region here in this epi bar, as we mentioned, is likely to be promoting tolerance. So there's significant cross-conservation with the T cell receptor facing amino acids of this peptide and Teraparatide with human parathyroid hormone, as you would expect, but also with tubulin, which is this a protein that's very prevalent and makes up the cytoskeleton in all of our cells. So the presence of this highly conserved, highly human epibar in frame 5 predicts that Teraparatide will be potentially tolerogenic and not immunogenic, and these are consistent with the clinical observations of only about 3% of the patients developing anti-drug antibodies. So again, now when we start to look at the in silico analysis of the impurities, where we did a few more than salmon calcitonin compared to the API sequence, we actually see that modifications to this N-terminal region, specifically within the 9 amino acids that form the epibar, we do see a drop in the Janus matrix score. So some significant drop, others kind of more of a minor drop. We also see changes in the overall epitope content based on those impurities, and those affect the epimatrix scores. And then we do have a peptide impurity here, which actually is a slightly lower epimatrix score, but a slightly higher Janus matrix score. So while we would expect these impurities here to be more immunogenic than the API sequence, we would expect this one to be about the same. When we look at these in vitro in the T cell assays, we again looked at these at 2 different concentrations, so one closer to the lower limit allowed by the ANDA guidance, and then equal to the API. At this lower limit, we really see no difference in the response to Teraparatide compared to the impurities, so we're going to focus more on the higher concentration. But what you can see is, as we mentioned, these are the impurities here that actually had a decrease in the Janus matrix score and a loss of homology, and we do see that each of these impurities was able to generate a response in more donors than compared to the API. This donor here, or this impurity here rather, that had a very similar epimatrix score and Janus matrix score, actually had about the same number of donors responding. So when you don't impact the human homology, the homology of the impurity, you don't get more of an immunogenic response. However, when you do lose tolerance or when you do lose homology, you actually see a pretty drastic increase in the number of responding donors. And this is all consistent with our in silico predictions. So the results of the IVIP T cell assay confirm the in silico results, predicting that the API will only be minimally immunogenic. Peptide impurities that cause a break in tolerance will be more immunogenic than the API. And in these assays, impurities were found to be more immunogenic than the API and in higher concentrations. They could result in a product that's more immunogenic than the RLD. So the concentration of the impurities relative to that of the API and the final drug product will also impact their contribution to the overall immunogenicity of the final drug product. And so we have our recommended path here. So we always want to start with the in silico analysis, looking at both your API and the impurities. And if we so we have a situation like salmon calcitonin, where the API is very immunogenic and non-human, or we have a situation where it's more human-like. So we want to look at those epitopes, and then we want to confirm that those predicted epitopes can actually bind in our HLA binding assays. If we find that we have epitopes that are predicted and that we confirm them in binding, we then want to go and confirm the tolerogenic or immunogenic nature of those in T cell assays. If we see no predicted binding, and if we predict no binding, we see no binding in vitro, we can make a fairly decent argument that you don't need to go into T cell assays because there won't be any epitope content. But here, where we do predict binding, we have binding, that's where you then want to go into these T cell assays to either confirm that's immunogenic or tolerogenic. So to recap, our in silico analysis will kind of help guide what's necessary in an in vitro assay. An immunogenic API, such as salmon calcitonin, is likely to have immunogenic impurities, and these impurities are unlikely to overshadow the immunogenicity of the API. However, when you have a tolerogenic API, such as Teraparatide, it will be minimally immunogenic. However, if any of the impurities present break that tolerance, then you can see an increase in immunogenicity, potentially to the RLD, if they're present at high enough of concentrations. And combining 2 of these methods, at least 2 of these 3 approaches, is very useful for determining the risk that of immunogenicity for your peptide and the impurities in the final drug product. What's next for immunogenicity risk assessment? We've developed a tool called the what-if machine that can actually predict process-related impurities and rank them based on their predicted impact of immunogenicity. And If you are interested in more of the what if machine, Amy Maddie will be presenting on November 4th at 4:15 on this new algorithm. So thank you very much to the EpiVax team, the FDA team, and our partners at Kubrick. This data was generated with the contract that we talked about earlier, but thank you to the whole EpiVax team, and thank you to all of you, and I will entertain any questions you might have.